Hello, everyone, and welcome to today's webinar titled Complex Primary Cell Assays, Strategic Tools for Biosimilar Comparability. My name is Sydney Panilla of LabRoots, and I will be your moderator for today's webinar. Today's educational web seminar is presented by LabRoots and brought to you by Rukin Bio. To learn more, visit rukin.bio. As a reminder, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You can also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Fiona Miller, BSc, Immunology and Pharmacology from the University of Strathclyde. Fiona, you may now begin your presentation.
Hello everyone and welcome to today's webinar. Thank you to LabRoots for hosting. I'd like to give a quick introduction to Rukin Bio. We are a preclinical CRO located in Scotland between Glasgow and Edinburgh. We are a team of around 110 scientists and support colleagues working across a range of therapeutic modalities. Our vision is simple: to become the most trusted research partner for biotherapeutic innovators. The company was founded in 2015, and in 2016 we launched our biosimilar characterization services. Since then, we've constantly run biosimilar characterization studies and also widened our areas of expertise to include translational immunology and cell and engineering. This means we can help support the development of both established and cutting-edge treatments. Since 2016, we've delivered hundreds of biosimilar binding and functional studies, supporting both early-stage development and formal analytical similarity packages. Our experience currently spans 18 biosimilar products, and we've generated data for both initial filings and in response to regulatory questions. To get us started, let's look at how we define a biosimilar. Biosimilars have been defined in similar terms by regulatory authorities. The FDA defines a biosimilar as a biologic product that is highly similar to and has no clinically meaningful differences in terms of safety, purity, and potency from an existing FDA-approved biologic, called a reference product. This means that the biosimilar must match the reference product in all aspects that affect patient outcomes. The EMA definition is very similar, stating that a biosimilar is a biological medicine highly similar to another biological medicine already approved in the EU, called a reference medicine, in terms of structure, biological activity, efficacy, safety, and immunogenicity profile. Both agencies require a rigorous, stepwise comparison, starting with analytical and functional studies, and where needed, clinical data to ensure that biosimilars are as safe and effective as their reference products. So while the wording differs slightly, exemplifying a lack of perfect harmonization, the core principle is the same. Biosimilars must be highly similar to the original biologic with no clinically meaningful differences. Biosimilars represent a complex challenge in terms of demonstration of similarity compared to generics. For small molecules, which are chemically synthesized, low molecular weight compounds, their structures are simple and well defined, so we can make exact copies. For biological molecules, large, complex proteins produced in living cells, their complexity and the nature of biological manufacturing mean it is impossible to make an absolutely identical copy. Hence, we have biosimilars, not generics. But even with all these differences in size, complexity, and the impossibility of making identical copies, there's one fundamental principle that remains true: structure still determines function. Whether it's a small molecule or a complex biologic, the biological activity and ultimately the therapeutic effects are dictated by the structure. That's why, in biosimilar development, so much effort goes into characterizing and comparing structure and function. It's the foundation for ensuring that biosimilars are as safe and effective as their reference products. So, what follows this foundation of assessments on the path to regulatory approval for a biosimilar? Here we have a summary of the current regulatory landscape for biosimilar approval. Since the first regulatory guidelines were introduced for biosimilars in 2004 in the EU, there have been many updates to bring us to the current regulatory positions. Starting at the bottom of the pyramid, the foundation of biosimilar development is comprehensive physical, chemical, and functional studies. These analyses use a range of orthogonal methods to compare the biosimilar and reference product across a wide range of quality attributes. The goal is to ensure that any differences detected are not clinically meaningful. Animal studies, once a routine part of biosimilar development, have now largely been phased out as a regulatory requirement. This reflects both advances in analytical technology and a better understanding of what constitutes meaningful evidence for biosimilarity. Next, clinical pharmacokinetic or PK studies remain essential. They are highly sensitive for detecting differences between products and are considered more informative than traditional clinical efficacy endpoints in many cases. Finally, while comparative clinical efficacy studies, or CES, have traditionally been required, especially for complex biologics like monoclonal antibodies, their actual value is increasingly being questioned. This is in part because there is a lack of suitable pharmacodynamic, or PD, markers that could serve as reliable surrogates for efficacy, meaning CES remains the default even when robust analytical, functional, and PK data are available. However, recent regulatory experience and analysis of biosimilar submissions suggests that, in most cases, the routine use of CES may not add meaningful value to the approval process. Let's look at some of that research in the next couple of slides. Here we have an illustration of the clinical development outcomes of 45 biosimilars assessed in the EU and US up to November 2019. Of these, 42 were eventually approved, with 38 undergoing comparative clinical efficacy studies. Notably, 100% of these CES confirmed efficacy, although in 3 cases it required some additional justification. In 2 cases, the outcome was that product improvement was required, but that was due to a lack of comparable immunogenicity. Therefore, the authors claim that for 36 of 38, the CES added no value to the scientific review process to approve the biosimilar. For the 3 biosimilars that were not approved, failures were attributed to non-comparable analytical and functional or PK data. Therefore, the authors go further to state that state-of-the-art analytical methods that include physical, chemical, and functional assays, along with PK studies, best inform efficacy outcomes, with no routine need for comparative efficacy trials. Taking a look at another study of this type, this analysis builds on earlier work by incorporating both clinical and quality data from 36 biosimilar programs submitted to the EMA between 2012 and 2022. This study focuses on the complex biologics, antibodies, and fusion proteins. Out of the 36 biosimilars, 34 were eventually approved, with analytical and clinical data supporting biosimilarity. 2 were not approved due to analytical similarity issues, even though they had overall positive clinical outcomes. 3 had PK-related clinical issues requiring repeat studies. 2 had CES concerns, but were still approved based on strong quality, PK, safety, and immunogenicity data. Crucially, the study found no instance where negative clinical data, including failed CES, led to a rejection, and no case where clinical trials were necessary to resolve residual uncertainties in the quality data. Here we see that biosimilarity can be robustly demonstrated in these studies through analytical and functional testing, supported by PK and immunogenicity studies without relying on CES. Overall, the data supports a refined clinical development pathway, one that prioritizes analytical rigor and PK evidence over traditional efficacy trials. To that end, earlier this year, the EMA released a reflection paper proposing a significant shift in the clinical development expectations for biosimilars. The new framework emphasizes a science-based, stepwise approach where the weight of evidence is built from physical, chemical, functional, and PK comparability rather than defaulting to CES or PD studies. This pyramid illustrates a streamlined hierarchy of evidence. Physical chemical analysis forms the foundation, ensuring structural and compositional similarity. Functional assays confirm that the biosimilar behaves similarly in vitro, reflecting its mechanism of action. Clinical PK studies validate comparable exposure in humans. The accompanying table summarizes how each study type is now viewed. CES and PD studies are no longer routinely required. PK studies remain essential. Animal studies are generally not needed. Functional and physical chemical studies are critical to inferring efficacy. The requirement for CES and PD studies was already flexible, but CES were typically conducted. Has this new proposal had a real-world impact yet? Pharmacon stated in February 2025 that based on intensive scientific dialogue with the US FDA, the Executive Board has concluded that the continuation of the trial is no longer necessary for the development and approval of FYB206 in the US. The therapeutic comparability of FYB206 with the reference drug Keytruda can be sufficiently demonstrated based on the comprehensive analytical data and the data from parallel phase 1 trial in patients with malignant melanoma. This really emphasizes the critical role of the structural and functional characterization steps in biosimilar development. What does that involve? One of the unique aspects of biosimilar development is that, unlike with new biologics, we start with a wealth of information about the reference product. This allows definition of an initial set of quality attributes even before the biosimilar product is fully developed. It is important that all plausible biological functions of the molecule, not just those considered clinically relevant, should be assessed. This comprehensive approach ensures that we capture the full spectrum of potential differences. As we progress with a detailed structural and functional characterization of the reference product, this helps to shape our quality target product profile, or QTPP. Also, throughout development, we refine our understanding of which attributes are most critical. These are the critical quality attributes, or CQAs. This criticality assessment considers both the potential clinical impact of each attribute and the uncertainty in evidence linking it to clinical outcomes. There are different approaches to criticality assessment, and regulatory guidance can vary slightly. For example, the FDA requires not only a criticality assessment, but also a tier assessment for each attribute, which then determines the appropriate statistical analysis. Once we have suitable biosimilar material, the analytical similarity assessment is performed. Methods to assess QAs need to be developed and their suitability for the intended purpose demonstrated, although full validation is not required. To show an example of just how extensive and rigorous the analytical similarity assessment is for biosimilars, we show here the wide array of attributes assessed for adalimumab biosimilars over the years, covering every aspect of the molecule's structure and function. Here we start with detailed physical-chemical characterization, including primary structure analysis, as well as higher order structure. We also look at post-translational modifications, like glycosylation patterns, using highly sensitive analytical platforms. Purity and impurity profiles are also scrutinized to rule out any differences that could impact safety or efficacy. Functional assays play a critical role in confirming the biosimilar interacts with its target, such as TNF-alpha, with the same affinity and kinetics as the reference product. Cell-based bioassays then demonstrate that the biological activity is equivalent, ensuring the biosimilar has the same downstream effects. Altogether, this is a very comprehensive suite of orthogonal methods. It's this level of scrutiny that gives regulators and clinicians confidence that biosimilars will perform just as safely and effectively as their reference products, even without CES. Building on this foundation, we now turn our attention to a particularly powerful subset of functional assays, complex primary cell assays. These assays bridge the gap between analytical characterization and clinical relevance by modeling drug action in a physiologically meaningful context. To explore that further, what are complex primary cell assays? Complex primary cell assays are in vitro assays using human primary cells or tissues that can be derived from healthy or diseased donors. They preserve native immune signaling and receptor diversity and are designed to model drug action in a physiologically relevant or disease context. They enhance biosimilarity assessments by providing mechanistic insight beyond standard cell lines, offering sensitivity to subtle functional differences, supporting claims of clinical relevance and safety, and are sometimes required by regulators for FC effector functional evaluation. Without CES, these assays potentially become elevated as the most physiologically relevant and complex assays to provide the assurance of bridging analytics with clinical outcomes. Regulators increasingly recognize that such assays can demonstrate functional biosimilarity in ways that simpler tests or even clinical trials cannot, thereby strengthening the totality of evidence. Let's pause for a moment and set out the topics for the rest of today's talk. To explore the role of complex primary cell assays in biosimilar evaluation, we'll look at this in the following sections. Firstly, ensuring functional similarity is a safety net for residual differences and how they can be applied as an investigative tool. Complex primary assays are specifically requested by regulatory agencies when effector function is part of the mechanism of action, which includes antibody-dependent cellular cytotoxicity assays, also known as ADCC assays. These assays can also help us to probe differences in physiological systems, especially when analytical or functional data raise questions about potential clinical impact. Next, we'll look at where assays can be used to support indication extrapolation. Complex primary cell assays can provide mechanistic evidence that supports the use of a biosimilar across multiple indications, even when clinical data is only available for one, or, as regulatory expectations evolve, in the absence of any CES. For example, they can demonstrate that the mechanism of action is preserved in disease-relevant cell systems, helping justify extrapolation to other approved uses. Finally, we'll discuss the use of assays for confirming the presence or absence of biological activity. Complex primary cell assays are essential for verifying whether a biosimilar does or does not have a specific biological function. For example, with IgG4 antibodies or engineered IgG1 designed to lack effector function, these assays provide the definitive evidence needed to confirm the absence of activity, ensuring that the biosimilar matches the intended safety and efficacy profile of the reference product. We'll keep coming back to these pillars as we move through the rest of the presentation. Let's now focus on the use of complex primary cell assays where effector function is designated as a critical quality attribute, or CQA, and how they can be used as an investigational tool. Effector function is often central to the mechanism of action for many monoclonal antibodies, and regulators specifically require that we demonstrate functional similarity in this area. When effector function is a CQA, complex primary cell assays, such as ADCC using PBMC or NK cells, are not just helpful, they're essential. These assays provide the most physiologically relevant way to confirm that the biosimilar matches the reference product in its ability to engage the immune system. But what exactly do we mean by effector function? Effector function refers to the biological activities mediated by the Fc portion of the antibody after it binds to its target antigen. This includes the recruitment and activation of immune cells or the complement system, leading to downstream effects such as cell killing or phagocytosis. On this slide, you can see a representation of the three main types of effector function assays. ADCP, antibody-dependent cellular phagocytosis, where antibodies promote the engulfment of target cells by phagocytes. ADCC, antibody-dependent cellular cytotoxicity, where antibodies direct immune cells like NK cells to kill target cells. And CDC, complement-dependent cytotoxicity, where antibodies activate the complement system to lyse target cells. Each of these functions can be measured to provide a direct readout of the biosimilar's ability to mediate these clinically relevant activities. There are many different ways to set up these effector function assays. Each decision from which target cell to use or effector population to select to how the data are analysed can influence both the sensitivity and the physiological relevance of the assay. At Bio, we don't just run standard protocols. We carefully design each assay to fit the specific molecule and regulatory question at hand. Data you see here are the result of that expertise. By making the right choices at every step, we're able to generate robust, reproducible, and visually clear datasets that give our partners and regulators real confidence in the comparability of their biosimilars. We won't go into every technical detail today, that could be a whole webinar in itself, but the key message is, great data starts with understanding the question being asked and making the right assay design choices. This is what we love to do at Bio. We've seen how robust assay design underpins high-quality data, but how do we decide which assays are essential for a given biosimilar? Let's take trastuzumab as a case study. For trastuzumab, ADCC is a key mechanism of action and is classified as a critical quality attribute. This table shows a structured risk assessment that was used to classify product quality attributes for SB3, a now well-established biosimilar. ADCC and Fc gamma R3A binding were both flagged as CQAs due to their high impact and moderate uncertainty. Importantly, regulators often expect more than one assay format to be used, combining highly sensitive methods with those that closely reflect physiological conditions, such as primary cell-based ADCC assays. This approach ensures that any potential differences are thoroughly investigated and that the biosimilar's functional activity is convincingly demonstrated, both analytically and in a physiologically relevant context. So, once ADCC is identified as a critical quality attribute, and we've seen how risk assessment guides assay selection, how do we ensure that the assays we develop generate data suitable for the rigorous statistical analysis applied for demonstration of biosimilarity? Regulators expect that assays used to evaluate CQAs are fit for their intended purpose and are supported by qualification data. Qualification is about demonstrating that the assay performs reliably and consistently, so that the data it generates can be trusted for decision-making. For complex primary cell assays that are used for key MOA in the similarity assessment, like PBMC-based ADCC, this means showing that the assay is accurate, precise, and specific for the intended measurement. We follow established regulatory guidelines such as ICH and USP to qualify our assays. The data shown here is from the qualification of a PBMC ADCC assay we developed for functional comparability of trastuzumab biosimilar and reference products. The data demonstrated accuracy of relative potency results within 10% across the target potency range of 50 to 200%. The dilutional linearity showed a strong correlation coefficient between observed and target relative potency results from 50 to 200%. An assessment of relative bias across the target potency range confirms that there was no inherent or trended bias. Taken together, The qualification results show that this assay demonstrates suitable performance characteristics for its intended purpose. The goal is to build confidence, not just in the science but in the data itself. When an assay is properly qualified, both developers and regulators can be sure that any observed differences are real and meaningful, and not just artifacts of the method. Let's look at how a PBMC-ADCC assay played a decisive role in the approval of ABP 980, a trastuzumab biosimilar, when used as an investigative tool. In the pivotal clinical trial, a higher proportion of patients treated with ABP 980 achieved a pathological complete response compared to those treated with the reference product, 48% versus 41%. This result meant that the primary endpoint was not met, as non-superiority could not be excluded. To address this, the applicant provided additional data using a PBMC-based ADCC assay, which is considered more physiologically relevant because it uses donor cells heterozygous for Fc gamma R3A 158VF. This analysis revealed that some Herceptin lots had a downward shift in ADCC activity, which was linked to changes in glycosylation patterns, specifically lower afucosylation and galactosylation, resulting in reduced Fc gamma R3A binding. These shifts in the reference product lots helped explain the more extreme results seen in the clinical trial, pushing the confidence interval beyond the pre-specified equivalence margin. By integrating this additional ADCC data with the totality of evidence from quality, non-clinical and clinical studies, the EMA concluded that biosimilarity was sufficiently demonstrated, leading to the approval of ABP 980. This example highlights that complex primary cell assays, such as PBMC-ADCC, can not only produce similar results to the CES, but also allow determination of the clinical significance of potential differences by providing information on the root causes of these differences. Now that we've covered how complex primary cell assays serve as a safety net for functional similarity, let's turn to the next major role these assays play: supporting indication extrapolation. Indication extrapolation is a key concept in biosimilar development. It's about providing scientific justification that a biosimilar can be used across all the approved indications of the reference product, even in cases where direct clinical data may not be available for every use. Complex primary cell assays are increasingly central to this process, as they can provide the mechanistic evidence needed to support extrapolation decisions. Let's explore how this works in practice. Historically, when a biosimilar developer wanted to extrapolate approval from one indication to others, regulators expected a clinical efficacy study in at least one sensitive indication, supported by strong scientific justification, including data from complex functional assays that reflect disease biology. However, as we have seen, regulatory expectations are evolving. The recent regulatory commentary now allows for the possibility that, with sufficiently robust analytical and functional data, including physiologically relevant in vitro assays, comparative clinical efficacy studies may no longer be required, even for the first indication. This shift potentially places even greater importance on complex primary cell assays. These assays are positioned as a critical bridge between analytical similarity and clinical performance, providing the mechanistic evidence needed to support indication extrapolation. In other words, as the reliance on clinical efficacy studies decreases, quality and relevance of our in vitro functional data become even more central to the biosimilar approval process. Let's look at how complex primary cell assays can support indication extrapolation using ABP 654, a biosimilar to ustekinumab, as an example. Ustekinumab is approved for a range of indications: Crohn's disease, ulcerative colitis, plaque psoriasis, and psoriatic arthritis. Its main mechanism of action involves blocking both IL-12 and IL-23 pathways, but the relative importance of these pathways and the cell types involved can vary between diseases. In the clinical development of ABP 654, the pivotal efficacy study was conducted in plaque psoriasis. However, to justify extrapolation to other indications, the developers used a panel of complex assays with PBMCs from both healthy donors and Crohn's disease patients. These assays measured key functional endpoints, such as interferon gamma and IL-17 release, and STAT3 or STAT4 phosphorylation in response to IL-12 and IL-23 stimulation, respectively. By demonstrating that the biosimilar matched the reference product in these disease-relevant functional assays, the developers provided strong mechanistic evidence that ABP 654 would be effective across all approved indications, not just the one studied clinically. This approach highlights the power of complex primary cell assays to bridge the gap between analytical similarity and clinical relevance, and to support regulatory approval for multiple indications based on a robust totality of evidence. To focus in further, let's look at a specific example of a complex primary cell assay used in the biosimilar development of ustekinumab. One of the key mechanisms of action for ustekinumab is the inhibition of IL-23-mediated T-cell activation, which is central to its therapeutic effect in diseases like psoriasis and Crohn's disease. To demonstrate functional similarity, we developed an assay using primary human cells to measure the ability of the biosimilar to neutralize IL-23-driven responses. This assay focuses on quantifying IL-17 production by Th17 cells in response to IL-23 stimulation, a direct readout of the drug's biological activity in a physiologically relevant system. Developing this assay required careful optimization to address challenges like donor variability and the low frequency of responsive cells. Over the next few slides, I'll touch on the development and application of the assay. To develop a robust and meaningful assay for IL-23-driven T-cell activation, we began by systematically assessing different stimulators to understand how they might synergize with IL-23 and produce a measurable response. This initial phase was data-rich and multiplexed. We used flow cytometry to simultaneously evaluate a range of cytokine responses, including IL-17, across different stimulation conditions. Donor-to-donor variation was also carefully considered at this stage, as primary human cells can vary widely in their responsiveness. While only data from donor A is shown here, this early screening allowed us to identify both the optimal IL-23 concentration and the most suitable donor for further experiments. Once we established the assay conditions, we transferred the endpoint to an ELISA format. This provided more consistent and controllable results than flow cytometry, making it ideal for a full comparability campaign. To ensure reproducibility, we reserved a whole leukopak from the preferred donor, cryopreserving enough PBMC vials to support the entire analysis. This careful, stepwise approach allowed us to generate high-quality, physiologically relevant data to directly compare the biosimilar and reference product in a system that closely models the drug's mechanism of action. Here you can see an example of the results from our comparative analysis, where we evaluated multiple biosimilars and reference products side by side using the final qualified assay format. To ensure that our results were both consistent and reliable throughout the entire analysis campaign, we established clear assay acceptance criteria during the qualification phase. These criteria were then rigorously monitored during sample testing. This approach allowed us to generate robust, reproducible data that regulators can trust. By maintaining high standards for assay performance and data quality, we help ensure that any observed differences are meaningful and that our submissions meet the expectations for biosimilar comparability. Ultimately, this level of rigor is what gives both our partners and regulators confidence in the conclusions drawn from our functional similarity assessments. In this context of indication extrapolation, and as we've seen with ABP 654 and Crohn's disease assays, access to a wide range of primary human cells, including healthy donors and cells from patients' relevant diseases, is a major advantage. At RicknBio, our access to these sources allows us to design assays that are more clinically meaningful and better reflect the biology of the target patient population. We maintain large banks of cryopreserved PBMCs from healthy donors, ensuring consistency and flexibility for assay development and qualification. We source fresh whole blood, including from genotyped donors, which allows us to tailor effector cell populations for specific assay needs and to model genetic diversity and immune responses. We can source PBMCs from patients with specific diseases, enabling us to test biosimilars in the most relevant biological context. We can often recall specific donors, allowing us to repeat or extend experiments with the same genetic and immunological background to improve reproducibility. This comprehensive access and characterization capability means we can deliver robust, reproducible, and clinically relevant data. When it comes to biosimilar development, especially for indication extrapolation, there's no set menu of assays to follow. Each program requires a carefully considered strategy tailored to the molecule, its mechanisms of action, and the regulatory questions at hand. Unlike originator products, which often rely on additional clinical trials for each new indication, biosimilar developers must build a much stronger case using in vitro and analytical data. This means you can't simply copy the pharmacology package from the reference product's original submission. This slide highlights that point using adalimumab as an example. On the left, you see the cell-based functional assays including in the original 2004 EMA submission for Humira, with complex primary cell assays highlighted in purple. On the right, you see the much broader panel of functional assays used in biosimilar submissions since then, again with complex assays highlighted. The expansion of the assay panel reflects both evolving regulatory expectations and the need to provide robust, mechanism-based evidence for biosimilarity and indication extrapolation. It's a reminder that biosimilar assay strategy must be proactive, scientifically justified, and always tailored to the specific development challenge. At RicknBio, we work closely with our mechanism of action and translational teams to make a broad range of primary immune cell models available to our biosimilar customers. This slide gives you a sense of the diversity of immune cell types and functions we can access and model in vitro, from dendritic cells and macrophages to B cells, T cells, NK cells, and regulatory T cells. This breadth is especially important as we look ahead to the next wave of biosimilars targeting checkpoint modulators like nivolumab, pembrolizumab, and abatacept. These therapeutics work by activating or inhibiting T cells, so having robust, physiologically relevant T cell models is critical for demonstrating biosimilar comparability and indication extrapolation. For these molecules, one of the most powerful tools in our assay toolbox is the mixed lymphocyte reaction, or MLR, a key assay for assessing T cell activation and modulation. On the next slide, we'll explore how MLRs can be used to evaluate the functional similarity of these biosimilars. On this slide, you'll see that there are two main types of MLR, one-way and two-way MLR. In a one-way MLR, monocyte-derived dendritic cells from one donor are incubated with allogeneic T cells from another donor. This setup provides a clear and defined system for assessing T cell responses. In a two-way MLR, PBMCs from two genetically mismatched donors are co-cultured, allowing both sets of T cells and antigen-presenting cells to interact and activate each other. This approach captures a broader range of immune effects. These assays allow us to capture a broad range of effects in a single experiment, including T cell activation markers, proliferation, and cytokine secretion. Regulators like the EMA and FDA expect functional assays to reflect the mechanism of action in the clinical indication. MLRs are a great example of this. They provide mechanism-relevant evidence in a relevant cell system, supporting both biosimilar comparability and the scientific justification for indication extrapolation. Before running a full mixed lymphocyte reaction study, it's important to pre-screen donor combinations to ensure you select pairs that will give you clear, interpretable results. On this slide, you can see how we systematically tested different combinations of T cells and monocyte-derived dendritic cells from 4 different donors in a two-way MLR. They are labelled A, B, C, and D. This creates 12 unique allogeneic pairings, each represented by a bar graph showing interferon gamma production at various concentrations of nivolumab. We use an interferon gamma ELISA on day 5 to measure T cell activation in response to nivolumab. The goal is to identify donor pairs that show a robust, dose-dependent increase in interferon gamma production. These pairs are then selected for the main MLR studies. This pre-screening step is crucial for optimizing assay performance, reducing variability, and making sure that any observed effects are due to the drug and not just donor-to-donor differences. It's another example of the attention to detail required to generate high-quality, mechanism-relevant data for biosimilar comparability. Here we see the results of our mixed lymphocyte reaction assay, used to evaluate the functional activity of 2 checkpoint inhibitors, nivolumab and pembrolizumab. Both of these molecules share a similar mechanism of action. They block the PD-1 pathway, thereby releasing the brakes on T cell activation. The diagram on the right illustrates this mechanism of action, which is also similar to the MOA for a number of other biologics that could be in the biosimilar development pipeline in the future. On the left, you can see the results for T cell proliferation and interferon gamma secretion. In both CD4 and CD8 T cell populations, treatment with either nivolumab or pembrolizumab led to an increase in proliferation, as measured by Ki67 expression, compared to the MLR alone or T cells alone. Similarly, interferon gamma secretion was dramatically increased in the presence of these checkpoint inhibitors, demonstrating robust T cell activation. This slide demonstrates how an MLR can also be used to evaluate the inhibitory effects of abatacept on T cell activation. The diagram on the right illustrates the mechanism. Abatacept blocks the co-stimulatory signal required for full T cell activation, effectively putting the brakes on the immune response. In this experiment, we first prime the system by blocking PDL1 and PDL2, which maximizes T cell activation and creates a strong baseline response. This sets the stage for a clear assessment of abatacept's ability to suppress T cell function. The bar graphs show the results. Both CD4 and CD8 T cell proliferation, as measured by Ki67 expression, are significantly reduced when abatacept is added to the primed MLR. Similarly, interfering gamma secretion drops dramatically in the presence of abatacept. This approach highlights the versatility of the MLR assay. Not only can it demonstrate enhanced T-cell activation with checkpoint inhibitors, but it can also robustly capture the inhibitory effects of molecules like abatacept. This is exactly the kind of mechanism-relevant functional evidence that regulators expect to see in biosimilar comparability studies for immunomodulatory therapies. In summary, combining optimized functional assays with thoughtful donor selection enables robust in vitro support for indication extrapolation in biosimilar development. Let's now move on to our final topic. Here we'll discuss the use of complex primary cell assays in understanding the physiological relevance of product attributes, specifically how these assays can help confirm or rule out the presence of a potential biological activity. Where there is even a minimal potential for biological activity in a biosimilar, it must be thoroughly assessed. This is precisely where complex primary cell assays offer significant value. They provide a more physiologically relevant context to evaluate such activity. Over the next few slides, we'll explore how these assays are applied to assess effector function potential and how the resulting data can inform and support regulatory decision-making. This slide highlights a common scenario in therapeutic antibody development, where effector function is intentionally reduced or eliminated. This is typically seen with antibodies based on IgG2, IgG4, or engineered IgG1 backbones designed to silence Fc-mediated activity. These design choices are often made for reference products to avoid unwanted immune activation, especially when effector function is not part of the therapeutic mechanism. However, the rationale for a lack of effector function must be carefully considered. In many cases, the target antigen is soluble, and the antibody is not expected to associate with the cell surface, making effector function biologically implausible. But this is not always straightforward. Some soluble targets like TNF have transmembrane variants. Others, like VEGF, can associate with the extracellular matrix, and other targets may be cell-associated through receptor binding. These nuances mean that assumptions about the absence of effector function should be supported by evidence, not just theoretical reasoning. This is where complex primary cell assays can play a critical role in confirming or ruling out residual activity in biosimilar programs. This slide exemplifies the challenge of demonstrating a true lack of effector function in antibody therapeutics. The data shown here were generated using a panel of monoclonal antibodies targeting CD20, each with common Fc region variants, including wild-type IgG1, IgG2, IgG4, IgG1 with LALA and LALA-PG silencing mutations, and the STR mutation developed by Mabsolv. The leftmost graph shows Fc gamma receptor binding assessed by SPR. As expected, the wild-type IgG1 demonstrates strong binding across the Fc gamma receptor panel, including CD16, a key mediator of ADCC. However, when we look at the IgG1 LALA variant, which is often described as Fc silent, we still observe measurable binding to several Fc gamma receptors. Similarly, wild-type IgG4 also shows some residual binding. In contrast, the STR variants are completely silent, with no detectable Fc gamma receptor binding. Translating these findings into functional assays, the middle graph shows ADCC activity. Here, both IgG1 wild-type and IgG1 LALA retain significant activity, while other variants, including IgG2, IgG4, and the STR mutants, show minimal or no ADCC. The rightmost graph, measuring CDC activity, tells a similar story. Only IgG1 wild-type drives a strong CDC response, while all other variants are essentially inactive. The key point is that commonly used silencing strategies, such as the LALA mutation or switching to IgG2 or IgG4 backbones, do not always result in complete loss of Fc gamma receptor binding or effector function. Without supporting data from complex primary cell assays to provide physiological context, these findings could raise concerns about residual activity. This underscores the importance of robust, physiologically relevant assays in confirming the absence of unwanted effector functions. In the case of ustekinumab, which targets the IL-12p40 subunit, there is evidence that this subunit may exist in a membrane-associated form on monocytes. For this reason, evidence of a lack of function was included in the ABP 654 biosimilar submission. The graph on the left shows data from our in-house approach. Primary monocytes were isolated from 2 donors and cultured with or without R848 stimulation to induce IL-12p40 expression. The results confirm successful stimulation, with both donors showing a marked increase in IL-12, IL-23p40 levels following treatment. As with all lack of function experiments, the inclusion of a robust positive control is essential. The graph on the right demonstrates the capability of our ADCC assay system. Using alemtuzumab, a CD52 targeting antibody known to mediate ADCC, we observe a dose-dependent increase in signal. This validation step is critical. It ensures that any observed lack of activity in test antibodies is due to true biological inactivity, not assay insensitivity. This slide shows the final application of the ADCC assay, testing biosimilar samples in our model with induced IL-12p40 expression. Having confirmed assay functionality in the previous slide, we now apply the system to evaluate actual test samples. The positive control demonstrates strong ADCC activity, confirming the effector preparation is capable of mediating an appropriate response. The dotted line represents the cutoff for positive or negative responses. In contrast, the test samples A, B, and C, alongside the non-binding isotype control, show no measurable activity. This supports the conclusion that these biosimilar candidates do not mediate ADCC against a physiologically relevant target cell. This type of data is critical for biosimilar developers when the reference product is not expected to engage effector function, but literature suggests potential cell-associated antigen expression. It provides direct evidence to support claims of functional similarity, or, in this case, functional absence. As we wrap up, I want to bring us back to the central theme of today's webinar, the evolving role of complex primary cell assays in biosimilar development. These assays are not just additional or supplementary, they're essential tools in the functional toolkit. They serve as a safety net for residual differences, helping to resolve uncertainties that might arise from analytical, functional, or clinical data. They provide mechanism of action specific evidence, which is especially critical when effector function is a designated CQA. They support indication extrapolation by demonstrating functional similarity across disease-relevant systems. And finally, they help us understand physiological significance, confirming or dismissing potential biological activities that could impact safety or efficacy. As regulatory expectations shift away from routine comparative clinical efficacy studies, the burden of proof increasingly falls on the quality and relevance of in vitro data. Complex primary cell assays are uniquely positioned to meet that challenge, bridging the gap between analytical similarity and clinical performance. At Rook & Bio, we believe that thoughtful assay design, access to diverse donor material, and deep mechanistic insight are key to delivering the kind of data that regulators trust and developers need. Before we close, I'd like to acknowledge the team who helped bring this webinar together. I'd like to thank Andrew, our Scientific Director for Biosimilars, for his research and content development. I'd also like to thank Amy for her work on the design and visuals. Special thanks also go to the wider team of scientists and study managers who contributed data and insights throughout. It's been a pleasure presenting today. Thank you for joining us, and I'd be happy to take any questions.
All right, wonderful. Thank you very much, Fiona, for your very informative presentation. We will now go ahead and move into the live Q&A portion of the webinar. As a reminder, if you happen to have a question that you would like to ask, please go ahead and do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. And in addition to our speaker, Fiona, I would also like to welcome Allison Kerr, Director of Customer Success, Rook & Bio. Okay, let's go ahead and get started here, ladies. Okay, the first question that we have, it says, at which stage in our biosimilarity assessment do you suggest that we run an MOA type of assay? Is there a particular stage where it brings best value? Can we wait to see if we get a request for it from regulators?
Okay, thanks, Sydney. I can pitch in on that one. Hopefully everyone can hear me. It depends, I guess, if the mode of action is part of the effector function, and as Fiona said in the webinar, in that situation you don't really have a choice. You really need to include mode of action style assays in your main submission to the regulators. If it's more to get an orthogonal evaluation over a wider range of assays, then you've got a bit more flexibility. We've been talking to customers for the last 2 years, probably, on mode of action assays. but we're seeing more inquiries coming through from customers, and we're now hearing that the regulators are asking them for this type of work as well. So it's no longer just a nice to have, and the caveat as is that depends on the biosimilar. So my answer and more customer facing role is the sooner the better. Come and chat with us. Certain assays maybe need access to different cell populations that are a bit harder to access or even expand in the labs. So there's never an obligation to do a particular assay with us, but we're always open to discussing it with the customers. So the sooner the better, really, to plan it in.
All right, perfect. We have another here that says, how easy are these assays to qualify? Can accuracy, precision, repeatability be established?
Yep, I can take that one. So really, the type of qualification depends on the intended use of the assay and the performance level of the assay itself. So if the assay is suitable for using in terms of reporting a quantitative result, then you must assess the level of accuracy, precision, and specificity to show that it's fit for purpose. And those qualifications will be performed according to the available regulatory guidance. It's entirely possible for these primary cell assays to be qualified like that. We've successfully qualified both PBMC and NK ADCCs and monocyte ADCPs in a relative potency reportable fashion. Really, any assay where you can achieve good signal to noise, a good level of variance, and a nice clear dose dependency of response should be suitable for qualification. Assays that don't quite have that level of performance can be qualified in a different manner. So they can be used in a qualitative manner in which you would identify samples as positive or negative. And in that way, you can qualify them in terms of ensuring suitable diagnostic sensitivity and specificity. So these assays can typically be qualified. The type of qualification really depends on your end use of the assay.
Perfect. Okay. The next one that we have here says, how many donors would you suggest we include? What is typical?
Again, as with all of these assays, I don't think there's an easy answer to any of these questions. The short answer is always kind of, it depends on what you're doing with the assay. So there are a number of factors that you would want to think about in terms of the number of donors. So in early development, you might want to include quite a large panel of donors so that you can understand how the donor population might vary in response to that assay. And then from there, you can select down to a smaller pool of donors for later work. And for example, in qualification, you might want to qualify maybe 3 donors to understand the precision across different donors. And that way you have a pool of donors available for you to use during sample testing. For assays where you're reporting relative potency and you want things to be really precise and repeatable, it would be ideal to limit that to only a single donor and then bridge in new donor material as is required. That's easy if you have cryopreserved leukopaks because you have a large quantity of cells that are always in the same state at the start of an assay. But it's more difficult to manage when you're using, for example, fresh blood donations because the donor's circumstances, donation to donation, might be different. So again, it really does depend on what you are using the assay for.
All right, wonderful. It looks like we have time for one final question here, so we'll go ahead and wrap up with this one. This next question says, can you prepare banks of isolated immune cells to reduce variance?
Yeah. I can answer that. And again, it comes with a caveat, a bit like Fiona's just mentioned. It depends on the cell type. We run a lot of NK ADCCs for biosimilar customers, and brilliant, why don't we create banks of NK cells so they're ready to go? And our experience for that particular cell type, when we put them through the cryopreservation process, become a bit more hyper-stimulated. So we see a higher background when we use them in an assay than if they were a bit isolated. So we know for NKs it's not great. However, for T cell assays, they respond really well there, and we've managed to expand different populations of T cells. We've got antigen-specific T cells we've expanded, so those are more ready to go. So yeah, it's possible. It depends on the cell type, I guess, would be my answer.
All right, perfect. Well, thank you again, ladies, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Rukin Bio, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time, thanks so much, everybody. Goodbye.